You learned in the last lesson that most traits are determined by genes. Genes for a trait can come in different varieties called alleles. For example, eye color is a trait that is determined by your genes. There are different alleles or versions of the gene for eye color, like blue, brown, green, or hazel. Similarly, hair texture is another trait for which there is a curly hair allele and a straight hair allele. Some alleles are dominant over other recessive alleles. That just means that a dominant allele covers up a recessive allele. A recessive allele may be hidden by a dominant allele. Dominant alleles are represented by capital letters, while recessive alleles are represented by lowercase letters. For example, going back to our eye color example, if brown eyes are dominant over blue eyes, you might use a capital B to represent the brown allele and a lowercase b to represent the blue allele. Any letter could be used to describe eye color. I just chose B in this case. The brown eye color, being dominant, could mask or cover up the recessive blue-eyed trait. For each trait, an individual inherits two alleles, one from their mother and one from their father. The combination of two alleles from the parents are the individual's genotype. A genotype is expressed with two letters. If the two letters are the same, for example, big R, or little r, that individual is called homozygous or purebred. If the two letters of a genotype are different, like big R, that individual is called heterozygous or hybrid. Remember that the capital letter is dominant over the lowercase letter, so a heterozygous individual would show the dominant trait while the recessive trait was hidden. The only way to display the recessive trait would be to have two recessive alleles. The physical appearance of a trait in an organism is called its phenotype. A phenotype is determined by the genotype or combination of alleles. For instance, an organism might have the genotype big T and be tall, or little t and be short. Tall and short are the phenotypes, determined by the genotypes big T or little t. If you wanted to predict the genotypes or phenotypes of offspring that a pair of individuals would have, you could use a tool called a Punnett Square. Punnett squares are tables that show all the possible outcomes of a genetic cross. They also show the probability of getting a particular outcome. Let's do an example using the flower color of pea plants as our trait of interest. Purple flowers are dominant over white flowers, so let's use a capital P to represent the purple allele and a lowercase p to represent the white allele. A homozygous dominant individual would have the genotype big P, and the phenotype purple flowers. A homozygous recessive individual would have the genotype little p, and the phenotype white flowers. A heterozygous individual would have the genotype big P, and, since purple is dominant over white, its phenotype would be purple flowers. Notice that the only way to show a recessive phenotype is to have a homozygous recessive genotype. Let's use a Punnett square to predict the outcome of a cross between a homozygous dominant plant and a homozygous recessive plant for flower color. Our cross can be written like this: big P times little p. We start by putting one individual on the left side of the box. Let's use the homozygous dominant individual and say that's the mom. Mendel's law of segregation states that when this female plant makes eggs, the two alleles for the trait will separate, so each egg receives only one allele. Therefore, we must split the two letters and put one letter next to each box on the left. This will represent that half of the plant's eggs will have this P, and the other half will have this P. Next, we'll place the second individual, the homozygous recessive male, on the top of the box, splitting the letters so we have one on top of each box. This represents the fact that when this plant makes sperm, half of them will have this P, and half will have this P. Because according to Mendel's law of segregation, each sperm only gets one P. Now that we have completed the setup for this Punnett square, let's fill in the table to see what genotypes could appear in our offspring. If this egg and this sperm were to fuse, the offspring would have the genotype big P, little p. If this egg and this sperm were to fuse, the genotype would be big P, little p. This egg and this sperm uniting would produce a genotype big P, little p. This egg and this sperm would produce an offspring with big P, little p. After filling out the Punnett square, we can see that all the offspring from this cross would be heterozygous and show the dominant purple flower trait, since the recessive white allele is covered up. Therefore, the probability of getting a heterozygous purple-flowered pea plant from this cross is 100%. 100% of the offspring would have the genotype big P, little p. 100% of the offspring would be purple-flowered. Let's do another example by crossing a heterozygous individual with a homozygous recessive individual. The cross in this case would look like this: big P, little p, times little p, little p. The heterozygous individual can go on the left side of the Punnett square. According to the law of segregation, half of this individual's gametes would have big P, and half would have little p. The homozygous recessive individual will go on top. We separate the letters to show each gamete has one little p due to the law of segregation. Now to fill in the table. If this egg and this sperm fuse, the resulting offspring would have the genotype big P, little p. This egg and this sperm would result in an offspring with big P, little p. This egg and this sperm would produce an offspring with little p, and the union of this egg and this sperm would produce an offspring with little p, little p. Upon examining the results, you can see that 2 of the boxes have the genotype big P, little p, and 2 have the genotype little p, little p. The genotype ratio is 2 to 2, which can be reduced to 1 to 1. That means that for every one individual with the genotype big P, little p, we will have one individual with the genotype little p, little p. We could also represent the same probability using percentages. 50% of the offspring from this cross would have the genotype big P, little p, and 50% would have the genotype little p, little p. The phenotype ratio describes what the flowers would look like. Half of the offspring from this cross have at least one dominant allele for purple flowers, so 50% of the offspring would have purple flowers. The other 50% are homozygous recessive, and so would show the white-flowered phenotype. That means that the phenotype ratio would also be 1 to 1, or 50% purple and 50% white. Let's do one final example crossing 2 heterozygous individuals, big P, little p, times big P, little p. We place one individual on the left side and one individual on the top of the square, separating the letters to show that the gametes these individuals produce each have only one P because of Mendel's law of segregation. Half the gametes would have a big P and half would have a little p. The union of this egg and this sperm would give us the genotype big P, big P. If this egg and this sperm fused, we would get a genotype big P, little p. This egg and this sperm would produce the genotype big P, little p, and this egg and this sperm would produce an individual with the genotype little p, little p. This cross gives us quite a variety of genotypes and phenotypes in the offspring. We have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive individual. This is a 1 to 2 to 1 ratio. This ratio tells us that for every homozygous dominant offspring we have, we should see 2 heterozygous individuals and 1 homozygous recessive individual. We could also say that 25% of the offspring from this cross should have the genotype big P, big P. 50% should have the genotype big P, little p, and 25% should have the genotype little p. The phenotype ratio would be 3:1, meaning that we would have 3 purple-flowered plants for every 1 white-flowered individual. This could also be written as 75% purple and 25% white. It's important to remember that these results are a probability. That is, what will probably happen, not necessarily what will happen in reality. We could perform this cross and get all white offspring. Is that probable? No, but it is possible. So far, we have been using Punnett squares to examine only one trait at a time. This is called a monohybrid cross. The prefix mono means one. Now I will show you how to use a larger Punnett square to consider 2 traits at a time. The prefix di means 2. So when we examine 2 traits, it's called a dihybrid cross. Let's examine 2 traits in fruit flies: eye color and body color. Red eyes are dominant over sepia-colored eyes. So we use a big R to represent red eyes and a little r to represent sepia eyes. It's important to note that you have to be consistent with your letters. So when I chose R to represent the eye color trait, I was consistent, using a capital R for red eyes and a lowercase r for sepia eyes. Oftentimes I see students try to change the letters and they'll use a capital R for red eyes and maybe a lowercase s for sepia eyes, but you can't do that. When you choose a letter to represent a trait, stick with that letter. So we have a big R and a lowercase r. That R represents the allele for eye color. Moving on to body color. Brown body is dominant over yellow body. So we'll use a big B to represent brown body and a little b to represent a yellow body. Again, we're being consistent with our letters. We choose one letter, B, to represent body color. Capital B for the dominant trait and a lowercase b for the recessive trait. Let's say we are crossing 2 individuals heterozygous for both traits. Big R, big B, little b, times big R, big B, little b. According to Mendel's law of segregation, the 2 alleles for each trait separate during meiosis. So gametes that these fruit flies produce can each only have 1 R and 1 B. So our next job is to figure out all the different combinations of R's and B's that the gametes can have. In the female eggs, we could have big R, big B, big R, little b, little r, big B, or little r, little b. Each of these gametes have only 1 R and 1 B. The same combinations of alleles could be found in the sperm of the male fruit fly. In order to conduct this dihybrid cross, we will need a larger Punnett square, one with 4 boxes across and 4 boxes down. That way we can fit the 4 allele combinations for egg and sperm across the top and down the left side. Filling in the boxes is pretty similar to the way we filled in the smaller Punnett square, except we are writing the genotype for 2 traits instead of 1. If this egg and this sperm combined, we would end up with the genotype big R, big B, big B. It's important to keep the R's and the B's together when we're writing the genotype in the offspring. This egg and this sperm would produce an offspring with the genotype big R, big B, little b. This egg and this sperm would produce big R, big B, big B. This egg and this sperm would produce an offspring with the genotype big R, big B, little b. This egg and this sperm, big R, big B, little b. This egg and this sperm, big R, little b, little b. We continue to fill in the boxes, making sure to keep the R's together and the B's together when we write the genotypes. Once we've filled in all the boxes, we can examine the phenotypes present. Any genotype that has at least one big R and one big B will have the dominant phenotypes: red eyes with a brown body. The boxes shaded in yellow have red eyes and brown bodies. There are 9 such boxes. In order to have the phenotype red eyes with yellow body, the genotype will need to have at least one big R and 2 little b's. These 3 boxes are shaded in green. There are also 3 boxes that have 2 little r's and at least one big B, giving them the phenotype sepia eyes with a brown body. These 3 boxes are shaded in blue. There is only one box remaining with the genotype little r, little b, little b. This individual has both recessive traits, sepia eyes and a yellow body. This dihybrid cross results in a Punnett square with the phenotype ratio 9:3:3:1. That is, 9 out of 16 individuals would probably have both dominant traits, red eyes and a brown body. 3 out of 16 would probably have the dominant eye color and the recessive body color. 3 out of 16 would probably have the recessive eye color with the dominant body color, and only 1 out of 16 would be likely to have both recessive traits, sepia eyes and a yellow body. The 9:3:3:1 phenotype ratio is typical of a dihybrid cross and shows the probable phenotypes of the offspring. It also nicely illustrates Mendel's law of independent assortment. You see, the eye color of the fruit fly had no effect on the body color and vice versa. Each trait was inherited independently of the other. So we could see a red-eyed fly with either a brown body or a yellow body, or a sepia-eyed fly with a brown body or a yellow body. The inheritance of one trait does not influence the inheritance of a second trait. This law of independent assortment holds true as long as the traits are found on different chromosomes. For example, in fruit flies, the gene for eye color may be found on this chromosome, while the gene for body color is all the way on this chromosome. Because these traits are on separate chromosomes, they are inherited independently of each other, as you just saw. Genetic linkage occurs when the genes for 2 traits are found on the same chromosome. 2 genes found on the same chromosome tend to be inherited together because they aren't separated during meiosis. This is why certain traits often appear together. For example, redheads often have freckles because the gene for red hair and the gene for freckles may be linked or found on the same chromosome. In fruit flies, the gene for eye color and wing length are linked or found on the same chromosome, so those traits might show some interesting inheritance patterns. In addition to genetic linkage, there are other exceptions to Mendelian principles. The principle of incomplete dominance explains why some traits show an intermediate phenotype in the heterozygous individuals. With incomplete dominance, the heterozygous individual shows a trait that is a blend of the 2 alleles. For example, a homozygous individual may have curly or straight hair, and the heterozygous individual has wavy hair, a trait in between the 2 extremes. Codominance is a phenomenon where more than 2 alleles exist for a trait, and multiple alleles are dominant. An example is found in human blood types. There are 3 possible alleles for blood types in humans: A, B, and O. Both A and B alleles are dominant, while the O allele is recessive. Someone with type A blood could have the genotype AA or AO. Similarly, someone with type B blood could have the genotype BB or BO. The only way to have type O blood is to be homozygous recessive for type O blood, having the genotype OO. The A allele and B allele are codominant, so if a person receives an A allele from one parent and a B allele from the other parent, then they will show properties of both and have type AB blood. Do you know your blood type? Another inheritance pattern that is different from those described by Gregor Mendel is polygenic inheritance. Many human traits are actually polygenic, or controlled for by many genes rather than just one. A good hint of whether a trait is polygenic is if it displays a range of phenotypes rather than just two. An example of a polygenic trait is skin color. Human beings aren't just black or white. There is a huge range of skin colors, ranging from ivory to tan to ebony, and everything in between. This trait is determined by many genes, and the combination of alleles you receive from your parents give you your own unique skin color. Height is another polygenic trait. People aren't just tall or short. We come in a range of sizes that are determined by multiple genes. Can you think of another human trait that shows a range of phenotypes and therefore might be polygenic? Genetics proves to be a very interesting field of study. Our knowledge of inheritance patterns and tools like the Punnett Square allow us to predict what our children and grandchildren might look like. More importantly, we can assess the risk of passing on genetic diseases like cystic fibrosis or hemophilia. By understanding how these conditions are inherited, we can hope to someday eradicate them with gene therapy and genetic counseling. If genetics interests you, maybe you could see yourself pursuing a career in this exciting field.